Hello again, everyone. I'm going to have two lectures on lipid metabolism. This first one is about lipid breakdown, and then the second one is about lipid biosynthesis. If anybody has their book, it's in chapter 15 and 16. So we're first going to talk about oxidative metabolism of lipids and liver and muscle. So essentially the breakdown of lipids in both liver and muscle. Fats are stored as triglycerides, which we've talked about. These are solid storage forms and transport forms of lipids. They're primarily found in the adipose tissue. So you have fatty acids that are esterified to a glycerol backbone. The major source of energy in the liver, muscle, and tissues is triglycerides, except for red blood cells in the brain. So when we think about the energy that's stored in our body, about 84% of it is fat, 15% protein, and less than 1% is carbohydrates. So the fatty acids in red and the glycerol backbone in blue there. So just as a brief outline, I'm going to go through an overview of lipid metabolism. I'm going to talk about fatty acids activation. So how are they activated for breakdown, specifically through this mechanism called the carnitine shuttle. And then we'll talk about beta oxidation in the mitochondria. So this is what occurs for most fatty acids, and there are some exceptions that we'll talk about as well. Then we'll talk about what ketogenesis is, catabolism of ketone bodies, and a little bit about regulation of lipid metabolism will be introduced here, but we will talk more about that in later lectures. So fats are transported through the body using chylomicrons. These are plasma lipid proteins. They contain phospholipids, cholesterol, proteins, and triglycerides. These fats are hydrolyzed by lipases by releasing the glycerol and fatty acids, which are then taken up by cells. At a high level, you have lipids being broken down into glycerol. Just change my pointer here. Being broken down into glycerol and fatty acids. Glycerol is converted to glyceraldehyde phosphate. That's converted into acetyl-CoA, and then this enters the TCA cycle. Fatty acids undergo beta oxidation, which we'll talk about, which produces acetyl-CoA, which can enter the TCA cycle. And importantly, it produces reduced nucleotides, FADH2 and NADH, which enter the electron transport chain, which we'll talk about in a later lecture. In muscle, acetyl-CoA is metabolized by the TCA cycle and oxidative phosphorylation to produce ATP, like we just mentioned there. In the liver, acetyl-CoA is converted to ketone bodies via ketogenesis. Here's a picture of ketone bodies. These are water-soluble lipid derivatives that are exported to other tissues, and we'll talk a little bit more about these. Fat metabolism is controlled by the rate of triglyceride hydrolysis in adipose tissue, and we'll talk again in later lectures about this regulation. It's regulated by hormones such as insulin, glucagon, epinephrine, and cortisol. Let's first talk about lipid metabolism in muscle. First step is fatty acid activation. So fatty acids don't exist in the free form in the body. We've talked about this previously. It either forms soaps or salts. It can be bound to plasma albumin in blood, or it can be activated to CoA derivatives using ATP. This is done through an enzyme called fatty acyl CoA synthetase, otherwise known as thiokinase, which catalyzes both reactions shown here. During this process, you consume one molecule of ATP. So even though this whole process is really to break down fatty acids to form energy or in the form of ATP, during this first activation step, you do consume one molecule of ATP. The fatty acid activation can be slightly different depending on how long your fatty acid chain is. Short and medium chain fatty acids cross the mitochondrial membrane by diffusion, so they are activated or undergo this first step in the mitochondria. Long chain fatty acids are activated in the cytoplasm and then transported via the carnitine shuttle, and we'll talk about that. And then very long chain fatty acids, we're talking about greater than 20 amino acids. This occurs in the peroxisomes. Let's talk about the carnitine shuttle. So again, for 12 to 20 carbon length acyl-CoAs, they're too large and polar to penetrate the inner mitochondrial membrane. So the carnitine shuttle allows transfer of a fatty acyl-CoAs into the mitochondria, where this beta oxidation occurs. Fatty acids are transferred from CoA to carnitine, which is just a small molecule formed by lysine and methionine, to form by carnityl palmitoyl transferase 1 in the outer membrane. So shown by this step here. So this is the outer membrane of the mitochondria. You get your fatty acid, CoA is attached to fatty acid, and then it's swapped for carnitine to form the fatty acyl carnitine. Then in this second step, carnitine acyl carnitine translocase in the inner membrane transfers acyl carnitine. So that's this step here, and you have this anti-transport mechanism. So free carnitine moves out and acyl carnitine moves in, shown here. CPT2 regenerates fatty acyl-CoA, releasing this carnitine, and then you can repeat the cycle. So essentially this is just a way to move the fatty acid into the inside of the mitochondria. Here's a little YouTube video to show you this in action if you're interested. I got a couple of YouTube videos from this person for some of these type of mechanisms. It's kind of helpful to see it drawn out. So once we have this activation, let's talk about beta oxidation in the mitochondria. So this oxidation of the beta carbon occurs. So the beta carbon here, and then this occurs in the mitochondria, and then you get cleavage in between the alpha and beta carbons. And so you release 2 carbon acyl-CoA units at each turn of the cycle. So from an 18 carbon unit, you get 9 acetyl-CoA units at each turn of the cycle. So the steps here are as follows. Essentially, it parallels the TCA cycle in that you have a dehydrogenation step shown here. So you get the dehydrogenation, then you get hydration, so you get adding of a hydroxyl molecule, and then you get dehydration again, you get the carbonyl formed here, then you have carbon-carbon cleavage by thiolase to form the remaining fatty acyl-CoA and acetyl-CoA, which can enter the TCA cycle. So again, I'm not going to ask you to draw structures here, but understanding the basic overall steps of dehydrogenation, hydration, dehydrogenation, and then the carbon-carbon cleavage, which the first 3 steps parallel the TCA cycle, is really what's important here. So during the process, 1 mole each of acetyl-CoA, FADH2, and NADH are formed, along with fatty acyl-CoA with 2 fewer carbon atoms. So that's all shown in this diagram here. The fatty acyl-CoA dehydrogenase is actually not a single enzyme. It's a family of enzymes with chain length specificity. So medium chain length fatty acyl-CoA dehydrogenases deficiencies can lead to some bad clinical outcomes. It can lead to low blood glucose from low concentration of ketone bodies. And then that can lead to high concentrations of medium chain carboxylic acid, acylcarnitines, and acylglycines in plasma and urine. It actually can be life-threatening because it limits gluconeogenesis and causes hypoglycemia. It can be treated by frequent feeding, avoiding fasting, and carnitine supplementation. So this is an important enzyme clinically, this fatty acyl-CoA dehydrogenase. Let's walk through beta oxidation of palmitate. So the net yield is 106 moles of ATP through oxidative phosphorylation, which again we'll cover in a different lecture here. During the process, 2 moles of ATP are invested at this initial beta oxidation step, which we talked about. We'll talk a little bit more about this later, but the ATP per gram of palmitate is 2 times greater than glucose because glucose is already partially oxidized. So that's when you see on the back of your cereal box or whatever, the calorie value of fats is about twice of that as sugars because fats are more reduced to begin with, so you can have less energy produced through oxidation. Here's a video about palmitate to walk you through that beta oxidation if you're interested. So those are the main pathways. There's some additional lipid metabolism pathways for long chain, unsaturated, odd chain, and branched fatty acids. And I'll walk through those. And what's important about these really is just understanding the differences from normal beta oxidation. I mentioned peroxisomal fatty acid catabolism. For these are oxidation for long, very long, and branched chain fatty acids. These peroxisomes are subcellular organelles that are found in all eukaryotic cells, shown by this little blob here. The enzymes in the peroxisome cannot oxidize short chain fatty acids, so they're really meant for these long chain and branched and very long chain fatty acids. The peroxisome is the principal site of production of hydrogen peroxide, and it accounts for about 20% of oxygen consumption in hepatocytes. It also has a role in production of acetyl-CoA for biosynthesis of cholesterol and polyisoprenoids, which we'll cover in different lectures. The actual beta oxidation step is very similar to the one in the mitochondrial pathway, but except for the acyl-CoA dehydrogenase is actually an oxidase rather than a dehydrogenase, which produces hydrogen peroxide in the process. So this is energetically less efficient, and it's not coupled to ATP synthesis. It's really to help in this hydrogen peroxide production. There are some clinical conditions associated with when you have defects in enzymes and peroxisome enzymes. The Zellweger syndrome, for example, has a defect in import of enzymes into the peroxisome. This can lead to severe multi-organ failure. It can lead to accumulation of long-chain fatty acids in neurons and lead to death at a very early age. Unsaturated fatty acids are already partially oxidized, so they have less calorie value, so less FADH and less ATP is produced. Again, the double bond, as we talked about last time, has a cis geometry, and it can occur at 3 carbon intervals. So essentially, if you look at this is an intermediate of beta oxidation, except for beta oxidation has a trans geometry. So essentially what you do here of unsaturated fatty acids is you can go through 3 cycles of beta oxidation to remove, and for example, for oleoCoA, you go through 3 cycles, and then once you get to this double bond here, you have an isomerase that takes it from the cis conformation to the trans conformation, and then you can just slide right into the remaining beta oxidation steps. So this is how our body deals with unsaturated fatty acids. What about odd chain? Odd chain undergo a beta oxidation just like even chains, except for you end up with an odd chain by the last thiolase reaction. So you end up with propionyl-CoA, 3 carbons instead of the 2 carbon units. Essentially what happens here is this is converted to succinyl-CoA by the 3 enzymes shown here, plus vitamins such as biotin and cobalamin. And then this succinyl-CoA can enter the TCA cycle. So again, what's important here is essentially our body just deals with this last step by converting it into succinyl-CoA. the name of the enzymes aren't terribly important. Branched fatty acids are a little bit different. These are some branched chain lipids that can be found in plant chlorophylls. So the beta oxidation is at a branch point, so it cannot be oxidized to a ketone. So essentially, you get a subsequent alternating release of acetyl-CoA and propanyl-CoA. And the acetyl-CoA can go to ketogenesis, and propanyl-CoA can make succinyl-CoA, as we talked about previously. Okay, what is ketogenesis? I mentioned in the muscle, acetyl-CoA is metabolized by the TCA cycle and oxidative phosphorylation to produce ATP. In the liver, this is a little bit different. Acetyl-CoA is converted to ketone bodies. So let's talk about ketogenesis. This is, again, a metabolic process that's unique to liver. Our body does this during fasting or starvation, and the fat-derived ATP and NADH concentrations in the mitochondria are high during this case. So the oxaloacetate-malate equilibrium is shifted toward malate. So then what happens is the malate enters the gluconeogenesis pathway, and the limited oxaloacetate results in limited TCA cycle activity. So the point is, you get through all this accumulation of acetyl-CoA, and our body finds something to do with that. The liver needs to get rid of that excess acetyl-CoA. Because CoA is essential for beta oxidation, we have a slowdown of beta oxidation in fatty acids, which is the primary source of ATP in the liver. So to recycle acetyl-CoA, to regenerate free CoA, this process is called ketogenesis. So free CoA is generated, and acetate ends up as acetoacetate, beta hydroxybutyrate, and acetone. And so these are what's known as the ketone bodies. So here's how the process works, the generation of ketone bodies from acetyl-CoA. So you get acetyl-CoA, releases free CoA for fatty acid oxidation. The process here involves synthesis and decomposition of HMG-CoA. This is through synthase and some lyase enzymes. So here's HMG-CoA. And then ultimately what happens is you get acetoacetate, and then this can be further processed into acetone and beta hydroxybutyrate, all of these which are ketone bodies. So we now have ketone bodies. We have free CoA released for fatty acid oxidation and acetyl-CoA recycled. But now our body has to do something with these ketone bodies. What happens here is the ketone bodies are transferred to the blood and taken up by tissues such as the skeletal and cardiac muscles. So for example, hydroxybutyrate can be converted back into acetoacetate, which can ultimately converted back into acetyl-CoA. This is increased in plasma during fasting and starvation and can be a source of energy. So it seems like a futile cycle because we're getting all the way back to acetyl-CoA, but the difference is here we're in a different tissue. The liver had to release that CoA, recycle the CoA for fatty acid oxidation, and then other tissues basically had to deal with the products produced by the liver. During starvation, our brain uses ketone bodies for about 50% of energy metabolism. It does this, by doing this, it spares glucose and degradation of muscle protein for gluconeogenesis. Here's some examples of typical plasma concentrations of fatty acids and ketone bodies during normal, fasting, and starvation state. So our normal plasma concentration of fatty acids is about 600 micromolar or 0.6 millimolar. In starvation state, fasting state, these go up, and the ketone bodies also go up during this time, and then our body can use these for energy through the cycle that I just showed on the last slide. Ketone bodies can end up in the urine, and this can occur with high fat, low carbohydrate diets and can cause metabolic acidosis, which is essentially just too much acid in the blood. I'm going to introduce regulation of lipid metabolism, but again, this will be covered in later lectures. But essentially, we have lipolysis, which is done through activation via glucagon and epinephrine hormones. So these activate lipases in the adipose tissue. The cortisol hormone actually has a similar effect, but it can be more chronic. So you get activation, these hormones activate the lipases to break down fatty acids in the adipose tissue. And these fatty acids are transferred through acylcarnitine, as we've talked about. And then beta oxidation of fatty acids in the liver can yield ATP for gluconeogenesis in the mitochondria. And in the liver, in the mitochondria, and in the liver, the acetyl-CoA is converted to and released as ketone bodies, as we've just talked about. And insulin can actually reverse this. So this is how lipid metabolism is regulated in the liver at a high level. We'll talk about more about global lipid metabolism in later lectures, as I've mentioned. I mentioned cortisol. There's actually syndromes associated with high concentrations of cortisol. This is a stress hormone. It can counteract insulin. It leads to, it stimulates gluconeogenesis, but when you have too much of it, it's known as Cushing syndrome, characterized by high blood sugar, muscle wastage, and redistribution of fat from glucagon-sensitive adipose depots to atypical sites like your cheeks, upper back, and trunk. This is just an overview slide that I'm going to come back to of how lipolysis and ketogenesis work together. But essentially, you can have, again, stored fats in the adipose tissue with triglycerides. You can break these down to glycerol and fatty acids. Glycerol goes through glycolysis. Fatty acids goes through either beta oxidation to produce acetyl-CoA or ketogenesis in the liver to produce ketone bodies. Ultimately, that can lead back to acetyl-CoA in other tissues. And then from this, you can go through the TCA cycle. You can build cholesterol off of this. We'll talk a little bit about that in later lectures. You can build steroids and bile salts from cholesterol. The energy transport chain from the oxidation products is to produce ATP. I'm going to have a whole lecture on this, so we'll talk about that later. So this just gives you a little bit of an overview of the whole process and how it relates to other processes in our body. And then the next lecture, this lecture, talked about lipid breakdown. The next lecture, I'm going to talk about how lipids are made.